I wanted to explain to you today is that antibodies are not only useful for diagnostics as we've used in this practical, but they're also increasingly important for therapy. And I've got here a list. You don't have to read this list in any detail, but just to see that there's many recombinant antibodies that are used now in the clinic for treating patients against many different diseases. Among those are cancer, autoimmune diseases, neurological diseases, and of course these are made by big pharma. Very often started in universities, but then frequently it goes to big pharma to make all of these antibodies. They can be either originally they were mouse antibodies that were used first. Now we're complete. You missed very little. I just mentioned that there are antibodies are not useful for diagnosis only, but also for therapy. And this is a list of monoclonal antibodies. Okay? Monoclonal antibodies that are used in the clinic. And the question is, of course, how can you make them? How can you use them? This is a slightly bigger list. This is a continuation of the other one. On the right, you see chimeric antibodies, humanized antibodies, fusion proteins, all kind of different recombinant antibodies, and I'm going to explain how they're generated, how they're made. What's the idea about it? They have complicated names. These ones you can just about read. Many of them unpronounceable, but there is a systematic name behind this, just as chemicals have systematic names, also monoclonal antibodies do. At least those that are used in the clinic. So, let's pick an example. Which one is the one that's used here? Yes, let's start here. The first syllable in these, let's pick one. The one that I cannot pronounce. Against B-cell leukemia, alemtuzumab. It was about right. The first one, infliximab, and so forth. So the first syllable in those antibodies are unique names. That really defines the antibody. Then it's the second syllable, for example, the lead in this example, it's for the field in which it is used. Neu, for example, is for neurological, because it was a neurological antibody. Sorry, the second, yeah, that's right. MAP is for monoclonal antibody. And then, depending on where the source, where it's taken from, if it's completely human, then there's a U in the name. If it's a murine antibody, so mouse monoclonal, for example, then there's a mo in the name. If it's humanized, and I'm going to explain to you what that is, there will be su. So they're the chimeric antibodies, half mouse, half human, will be xi. So this is the next example. Oh no, it's a complete list. So you can see that the source is on the right, source infix, human mouse, it can be rat monoclonal, hamster, so they all have a corresponding letter that defines it. If it's a neurological antibody, as I mentioned here, there it will be a neu in the name. If, what else do we have? A lead for the immune system, and so on. So each area of research has its own syllable, and after a while, if you're in this field, you might, from the name already, you know more or less what the antibody might be related to. You don't need to learn this. I'm not going to ask about this in the exam. Just want you to be aware about this. Okay? This just to have the proper structure in a presentation. So this is really something that I want you to know. We've discussed it in one of the journal clubs. So those of you that couldn't make it, then please also have a look at these domains. I think they're important because if you want to make antibodies and use antibodies, you need to think of the structure. So in total 12 domains, if I counted right, and each of them can be manipulated separately. When in reality they look like this. You can see each of the domains. Here's one. the purple residue there is the sugars that keeps the two heavy chains down there together. And you can see that the domains are kind of twisted around each other. So what is a chimeric antibody that I'm talking about? A chimeric antibody is when you have generated first a mouse antibody and you want to use it in humans, then it will be very immunogenic. So if you take a mouse monoclonal and wanted to that was directed against a cancer protein, then it would be very nice to use this mouse monoclonal in cancer patients. Well, if you do that, inject it once, it will work. It will help to treat the cancer, but you cannot use it the second time because the patients will mount an immune response against the mouse antibodies. And the second time they will get a high fever and maybe an allergic shock as a response to injection of an allergen, right? So you cannot do that. So the first step in improving these antibodies for clinical use was to replace all the constant part of the antibodies, shown here in blue, throw away the mouse sequences and replace them by the human sequences. For example, the human IgG1. Okay. Up there are all the variable sequences that you need for binding, so they will be remaining mouse. So these are the chimeric antibodies used clinically. I think they were abbreviated XI. Sorry. The next step of making these antibodies more human even is replacing there's in the variable domain there's also some parts that are rather constant. They're called framework regions. Those are the pieces up there in blue still. In between you have the CDRs, the complementary determining regions, and these are really the very variable parts of the antibody. So even in the variable domains, there are parts that are rather constant, and those you can then also replace by human sequences. So that makes such an antibody more human. And these antibodies are called humanized, as you can see in the top. The thing is that this is not an easy procedure because the framework regions that are shown up there in the top also determine the structure of the antibody in that area. So very often the antibodies will lose some affinity. So you need to mutate other amino acids to compensate for that. So this is a very tricky procedure to humanize an antibody without losing the activity. Okay. Next step to make it even more human is, of course, to generate completely human antibodies. So to start from an organism that has the human immunoglobulin genes, there are mice that have that, to transgenic mice that have, instead of the mouse antibodies, will produce human antibodies. Or you start off from a human patient that might have an autoimmune disease or might already have overcome an infectious disease and we want to clone the antibodies from such a donor. That's also possible.
The framework region, as I understood, is like the CDR region, or is that the variables?
Yeah, the CDRs are the complementary determining regions, that's the variable parts. The framework regions sit in between. Okay. So that's the blue parts there in the top, the small arrows.
The human parts.
Yeah, this is the framework 1, 2, and 3. I think we that all. And here's a short piece of constant framework as well. I think that one's called framework 4. I'm not quite sure about that one. So anyway, the CDRs, that defines the antibody binding. They are very variable, different between all antibodies. In the top you also have the framework regions. They are rather constant. Sometimes there are also mutations there. Not completely constant either. So how do you make fully human antibodies? Because those are not, or almost not immunogenic at all. So when you want to treat a patient, the best thing is to treat a patient with a completely human antibody. And this is a summary table where how you can generate those. So you can either get them from human plasma cells, you can get them from human B cells. All of those have rearranged immunoglobulin genes. You can use humanized mice, that's what I just mentioned. There are mice that have replaced in the germline, in the DNA of all nuclei, the mouse antibody genes by human antibody genes. So these are humanized mice. And there's also a DNA technology using phage display. I'm not going to talk about that. There you make the antibodies in bacteria based on human sequences. So they've used human donors, many human donors, to isolate the DNA, isolate all the sequences of different antibodies and put them all together in a tube with a huge variability of sequences, and then they select the best one. This is a technique that's called phage display. What we are using in the lab is one of these techniques. It's called B-cell immortalization on memory B-cells. And this is the following trick. There's a natural virus that infects our B-cells. That's the Epstein-Barr virus. Actually, more than 90% of us have this virus in our body. It integrates in B-cell in a stable way. When you're first infected, it's either asymptomatic or you develop a Pfeiffer's disease, which is mononucleosis. Anyway, this virus infects B-cells, and very occasionally it integrates in the DNA of the B-cell and will immortalize that. So then you get a kind of cancer cell line, which also can happen in humans, and it's Hodgkin's lymphoma, if I'm not mistaken. So not a non-Hodgkin's, but a Hodgkin's lymphoma, very easy to treat. It's not very dangerous. It's a chronic disease. In any case, you can do the same thing by incubating B cells from a patient or from a donor with Epstein-Barr virus, and then this might lead to an immortalized B cell clone. To make that efficient, you need a trick. You need to activate those B cells with a piece of DNA, CpG, so it's methylated DNA, bacterial DNA. And B cells have a receptor, a toll-like receptor, that can recognize bacterial DNA. So when you add that, these B cells will become activated and start growing, especially when they're immortalized with Epstein-Barr virus. What will happen then in the next step is that those B cells will start to divide and produce antibodies. B cells normally don't produce very many antibodies. Plasma cells do, B cells don't. They produce a few. But in this way, you will generate a monoclonal cell line. So how this is done? You use an ELISA plate. Not ELISA in this case, a cell culture plate, also 96 wells. You add between 1 and 50 B cells per well. You add the CpG, you add some growth factor, IL-2 or IL-6. And what you also need to add is feeder cells. So that's irradiated PBMCs, peripheral mononuclear cells. And this will allow the B cells to grow a little bit better in the first stage. So they don't like to sit all alone in a well. They need the environment of other cells to be surviving. You need to add the Epstein-Barr virus, and this is produced in a cell line. So it's a cell culture supernatant that we just need to add. Of course, this is a class 2 organism because it's very infectious to us as well. Most of us do have antibody titers, but of course there's a risk of developing Pfeiffer's disease when you work with this stuff. So you need to be a little bit careful. It's a safety class 2 organism. So only special labs are equipped to work with those stuff. Next step to see if there's an antibody production, and we use this a lot, is the dot plot in a very similar way as we've done yesterday. You can develop it with fluorescence like we've done yesterday, or you can also use peroxidase and use DAP, diaminobenzoate, that makes it precipitate on the nitrocellulose, and then you can really see it directly after incubating. It's another way of doing it. So this is an example of a dot plot. You can see some are negative. You can see here in the bottom there's a dilution series also as a control, in a similar way as you've done yesterday. It's a fast way of screening the clones. Of course, you could also use, since this is an ELISA plate form, you can also use a cell culture supernatant in an ELISA. It's also easy to transfer 1-to-1. It's also doable. And the next step, of course, is to screen all of the clones that you've generated for the activity that you want. I mean, there are lots of different antibodies produced there, so you need to find the right one. But there are two ways of making this a little bit more efficient. One is to upgrade this, to make lots of plates, not only one, but 10 or 20. in parallel and test them all. Or you can select already the B cells for the right activity. So that since they produce the antibody, you can do a fax to find the right B cells that will bind to your antigen. So you need a fluorescently labeled antigen to then bind your specific B cells. Next question you need to ask yourself, what kind of antibody isotypes do these B cells then produce? We did it with thymocytes. So also in the human thymus, in some diseases you will find B cells, the memory B cells. So we asked the question, what kind of antibodies are produced there? And some of them were IgM, others were IgG. And this differed between control thymuses from healthy people and from patients with myasthenia gravis, which we were interested in at that point. So this is one way of getting data out of those clones. So then the next question is, when you've got a clone, is this really monoclonal? Monoclonal meaning that all of the B cells produce the same antibody. Polyclonal meaning the B cells make different type of antibodies. The way that we can differentiate the antibodies is by looking at the length of the CDRs. They're not constant. They don't have a constant length, but they differ in their length between I think 10 and 30 amino acids roughly in most cases. And by doing a PCR between the framework regions, which you see down there, and they sit in between the CDRs, you can determine the length of the PCR fragment, and then you know if it's always the same, that is a monoclonal. So in the top you can see the results for polyclonal DNA from polyclonal B cells. You can see the length of the fragments that are produced by PCR varies a lot, because it comes from lots of different B cells. In the bottom you see an example of a monoclonal line that has one length for the framework 1, one distinct length for the framework 2, and one distinct length for the framework 3. If you see somewhere appearing 2 dots or 2 lengths, 2 different lengths, you know it's a biclonal cell, which it might also have. 3 would be triclonal, and after that I would call it polyclonal. So the principle of that is that different antibodies have different lengths of the CDRs. They have different sequences and they also have different lengths. And the next thing that you might want to do is a functional assay. In this case, we were curious whether or not these antibodies that we generated bind to the muscle. You can do the same thing by testing if it binds to kidney, brain, whatever organ you're interested in. And the next step is, of course, not to look at the whole tissue, but to look at individual antigens. And then you would need an ELISA or a Western blot or something like this to test if your antibodies are binding. Again, this is just an example of what you might want to do with such antibodies that you've generated. If you want to know something about the actual sequence of the antibodies, well, you need to sequence the DNA or the corresponding RNA that's produced in the cells. So what we do typically is we use primers against the framework regions, and then we amplify the DNA, the cDNA, and then we sequence it through. And then we look at the mutations in the antibody genes as compared to the germline. Maybe I could recommend to you, if you're interested in the background, I'm not going to ask in the exam, but I think it's important to know some basic immunology. Look up the way that B cells rearrange their DNA, and then you will read about somatic hypermutations and so forth, and rearrangement of VDJ fragments. I think this is rather important. So how do you analyze the results? Most genes you can just blast. It's very easy. You will find very fast what kind of protein or gene you're looking at. With antibodies, slightly more difficult because they're extremely variable. I mean, they're designed to be variable. So if you want to compare antibody sequences, you can use from the NCBI website and the IgBlast database to compare your antibody sequence to those that have already been described. Just want to point out that there's something like that. What we were looking at is in So these are results from our sequencing analysis. If the immune response that was going on in the thymus was monoclonal, polyclonal, or oligoclonal, and in this case it was oligoclonal because sometimes we found the same sequence again. So that means that there was an immune response going on in the thymus, because sometimes we find exactly the same sequences. So those clones were representative more often, so that's why we get what's called clonal expansion. Same thing that happens after the Epstein-Barr virus immortalization is that some clones that are activated will start to grow and to divide. And then you can find them back multiple times. So what this really means, we're not sure, but we find it more in the thymiform myasthenia gravis patients. So that's probably why this organ is involved in the autoimmune response. You can also compare the different sequences with the software. This is one example. So this makes a phylogenetic tree. So you know how diverse and how these antibodies have developed by adding mutation on mutation, so you know where they really come from. If they're very similar, you will see like up here, so when they're grouped together like this, they have differentiated highly, so the chance is very high that they've encountered their antigen and tried to optimize their binding by making different mutations of this version. If they're very dissimilar, these are just parallel immune responses against different antigens. So if you wanted to make such an antibody yourself, that's the next step. If you decide one of these antibodies is really interesting, I want to make this into a therapy, I want to produce lots of it.
Can I ask you a question?
Yeah.
Like you now check you for either if there are monoclonal or polyclonal or oligoclonal antibodies are produced. Do you check them for you could have different B cells making different clones of antibodies, but also one antibody could be specific for different regions, so therefore so can you maybe enlighten that? Do you answer that question?
Not quite. Okay, explain more.
Do you check for whether there are more B cell lines in one well, which would be then producing different antibodies? Or
that's the question if it's monoclonal or not.
Yeah. But it could also be that one antibody would bind in different ways to an antigen, right? So and therefore
if it has exactly the same sequence, it will bind always to the same antigen.
Yeah.
So the binding sequence in this case, we've only looked at the CDR3. You also need to look at the others. If those sequences are completely identical, it will always bind to exactly the same antigen. So that those CDR3s, they define the antibody and the antigen. So if you have sequenced this, you can completely predict what this antibody is going to do. Well, not predict, you need to measure it, but if you know this is the sequence, then you know that it will always bind the same thing. So if you have that sequence, you can make recombinant antibodies. And one way of doing it is by having the genes synthesized by a company such as this one. So you send your DNA sequence, for example, all the nucleotides in the right sequence to this company. They will send you back a plasmid, and in this plasmid you will have the variable domain of your antibody. The next thing that you need to do is to clone the corresponding DNA, the corresponding gene, in an expression vector that combines it with the constant part of the antibody. And such an antibody vector could look like this. This is one that we're using for making antibodies against A-beta in Alzheimer's disease, for an Alzheimer immunotherapy. This is a 3D6 antibody. There's already one that's used also clinically. And we're using it to make two different antibodies with exactly the same binding properties. Both are directed against A-beta, against amyloid beta in Alzheimer's disease, which is aggregated. And we want to make different isotypes of it to look at the effector functions. What you see in such a vector is that this is actually not quite correct. This is a different vector. Anyway, One possible way of doing is to put the genes for the heavy and the light chain in the same vector. You can also use different plasmids and co-transfect them. You will need a promoter. In this case, the plasmid contains 2 CMV promoters for both the heavy and the light chain. CMV is cytomegalovirus, so it's a very strong promoter that will work in any eukaryotic cell. So in CHO cells and HEC cells. Even if you would transfect it into your muscles, they would also produce antibodies, which they normally don't do, of course. You will also need a way to produce this plasmid, so that's why these plasmids are designed to be shuttle vectors. They will also grow in bacteria. So you can use this plasmid both in mammalian cells as well as in bacteria. In bacteria, the plasmid will just grow, and in mammalian cells, they will produce antibodies and not grow. So you need to include in your plasmid the 2 expression cassettes. One is for the light chain, and one is for the heavy chain. And as I said, alternatively, you could co-transfect 2 different plasmids separately, one for the light chain and one for the heavy chain, and transfect them into a eukaryotic cell line. These cells don't have to be these cells, they can be any kind of cell. If it's a mammalian cell, even an insect cell will produce antibodies for you. So you can easily put the human antibody genes in a mouse cell line, or vice versa, the mouse antibody genes in a human cell line. That will work perfectly. So I just mentioned here, we're looking at the same antibody in terms of the Fab arm, so it will bind the same antigen, in this case A-beta, and they produce different antibody isotypes. And why is that? Because different antibody isotypes have different effector functions. So this is a scheme taken from the Janeway Travis textbook. You can find it in the library if you want. You don't have to, but it's in a very good immunology textbook. And I think, oops, it's not in there. What you can see is that they have pluses and minuses, and in the different respective effector functions. So what they can do is, for example, they can activate NK cells, they can activate the complement system or not, and in that way they differ a lot. So some antibodies are very good in going through the mucosa. For example, the IgA antibody you don't find so much in the blood, but mostly in mucosa, in the gut and so forth. And others, IgG, you will find in the blood. You'll see that the concentration range of these antibodies also differs a lot. For example, IgG1 you'll find in a concentration of 9 grams per liter, or 9 milligrams per milliliter if you prefer that unit. So that's 10 times more concentrated than the antibody that I gave to you for the dot plot of the ELISA. So your blood is very highly concentrated in IgG. And then you also find other isotypes. For example, the IgG3 is more or less 10 times more diluted than the IgG1 in your blood, but you can also detect it. So if you're interested in the effector functions, because or we are interested in it because they will define if an antibody will, when it binds, activate the immune system further, will activate the complement system, for example, lead to inflammation or not. So what are the antibody effector functions? Well, this is one of them. So if you see on the left that an antibody binds typically to a pathogen, a bacterium, for example, it can also just bind to or a yeast cell, something happen to be infected with Candida or something similar, or Aspergillus. Then the antibodies will bind. The next thing is that the cells of the immune system will recognize that a high density of antibodies have bound to a surface, and then they will be activated and try to kill the cells that the antibodies have bound to. So this is antibody-dependent cytotoxicity. That's the first one. The second mechanism is antibody-dependent complement lysis. So this is when antibodies, so it doesn't need any cell, it just happens by means of proteins that are in your blood. And what they will ultimately lead to, can I use the mouse, is the formation of pores and membranes. So the antibodies are kind of shooting holes in in membranes of cells. Of course, also thought to kill bacteria. It's a kind of complex mechanism involving 9 different proteins of the complement system, C1 to C9, and the ones that are involved in the pores. Where's my mouse? The ones that are involved making these pores that you can see here are the proteins C5, C6, C7, C8, and C9. And the complex that is formed here is abbreviated with C5b9. It's a fragment of C5, and moreover C6, C7, C8, and C9. And mostly C9 forms these transmembrane pores. The hole in the cell is, of course, very bad news for the cell. It's kind of shot. It will die because it will lose all the proteins inside. It will lose the ions inside. Lots of calcium gets into the cell. Typical cell will die from this. Also bacteria, they don't like it. So this is what antibodies can do when they bind in a high density. So this is a scheme of what happens when antibodies bind to the surface. In this case, also in autoimmunity, the same thing goes on, unfortunately, because this is directed against bacteria normally. So this is the first protein to bind, the C1q, just schematically. It's also present in your blood. And when the antibodies are in a high density, then the C1q will via C4 and C3 activate C5b9. And C5b9 is this green complex that I've represented there. It really goes outside of the membrane. You can see it here in the previous picture. Just want to show it to you. There's this complex from the side. You can really see that it sticks out as a short sort of tube outside of the membrane. So back to this one. Well, what will happen then in a typical cell, also in a neuron? For the neuron is particularly bad because it will lose the normal gradient of ions that are present there. So the potassium that is normally inside of the cell will leak out. The sodium and the calcium will get inside of the cell. So this is a functional impairment of the ion gradient that is keeping the cell membrane functional, and the calcium, of course, can induce apoptosis. There's another effector mechanism antibodies have. They can cross-link, and I think I've shown this slide to you before. They can cross-link to receptors, and they can modulate it, and then you get this internalization of the membrane and the antigen. So this doesn't involve the lysis of the cell in any way. It will just lead to less proteins on the surface. Now, when you think about application in cancer, this is not something that you want at all, because in this way the antibody just gets inside of the cell, leaves the cancer cell viable, but just has less of this antigen on the surface, which makes it then inert to the immunotherapy. So that's a bad thing to happen in cancer. It's a bad thing to happen in autoimmunity, but only if the antigen is needed. So this is not something that always happens. This doesn't happen if the antibodies are monovalent. If one side of the antibody is binding and the other one is not, for example, by knocking out one of the complementary determining regions, then you can make, in a recombinant fashion, an antibody like this. So this was a trial that we had a couple of years ago. We wanted to make an antibody that looks like this. We wanted to knock out the capacity to activate the complement system that you can do by mutating a couple of amino acids here in the Fc part. We wanted to knock out the binding on one of the heavy chains, and we added a flag tag and a his tag to differentiate it to heavy chains. So this you can all do by changing the plasmid that encodes for your antibody. Of course, you need the sequence, otherwise you can't do it. So this is an example of a recombinant antibody. And why did we make it? Because we wanted to generate an antibody that is monovalent, so one that only can bind with one side. Because typical antibodies can bind with both arms and cross-link antigens. Now this is how you would make such a monovalent antibody. You would need to transfect two different plasmids because you have two different heavy chains, and you co-transfect them into an expression host. In this case, we use CHO cells. It's a mammalian cell line. Chinese hamster ovary cells. They can produce antibodies at a high rate. but other cells like X cells work even better. This is my last slide, just to show that antibodies do not always cross-link. And one antibody that doesn't is a human G4 antibody. And it's a funny thing. The B cell will, that encodes for such an antibody will always produce a cross-linking antibody initially. Another B cell will produce another G4 antibody. And what happens in the blood, quite fast, and these antibodies fall apart into half molecules composed of one heavy and one light chain. And the other antibody will do the same. What happens is they can recombine to make these hybrid antibodies. And since they have two different antigen binding sites, they will not cross-link the same receptors. So, for example, the original antibody will cross-link, but the resulting antibody very fast after that will not. So this is something that happens specifically with the G4 antibody. And of course, this stops the effector function of cross-linking of this antibody. And we're currently investigating what the biological relevance is of this FAB-arm exchange that is going on right here. Because the cross-linking as an effector function is not something that is generally considered very much, but we think it's important. So, was my last slide anyway, I think? Yes, no, this is not relevant. I leave it at that. You can think about possible ways of analyzing in any assay if such hybrid antibodies are present, if you like, as a thought, and how to analyze if there's an antibody that binds against one receptor here and another receptor on the other membrane. So these are assays that we're currently trying to optimize. So if you have any ideas of that, you're welcome to think about it. Yeah?
Yeah, I have a question about the monoclonal or the polyclonal part.
Yes?
Because I don't think I really understand what it means right now.
Okay. So a monoclonal antibody is an antibody that is derived from the same type of B cell. So all the B cells that I'll explain on them. So the question was, what is the difference between a monoclonal B cell and a polyclonal B cell?
Yeah, but then it would be a characteristic of the B cell itself, right? If it produces the same antibody or a different antibody.
Yes.
What I understood was that a polyclonal antibody can bind to different stuff.
Okay. I think that's. So first of all, if you have a B cell that produces an antibody that is directed against target A, yeah, and this one divides into two B cells, that will still produce, of course, because it has the same DNA, the same rearrangement, antibodies against A. So this all is a monoclonal cell, like, because all of these cells are identical. One comes from one clone. What could happen at some point is that there is in the germline here a mutation. So then this antibody would be directed against something else. Right? Then it's directed against B. So now, if you have these together in a well or in a human, this is not monoclonal anymore. These are two different clones. And if this diversifies further, you will get a whole range of different antibodies. If this one, and because these antibodies are different, they will recognize different things. So this is a different this is the definition of polyclonal.
Okay, but one antibody can only recognize one specific region of something.
Yes.
So just one antibody cannot be polyclonal.
No, exactly.
It's just polyclonal antibody because two antibodies are different.
Yeah.
So if you look at all the antibodies in a human or in a goat for that matter, they come from lots and lots of different B cells directed against A, B, C, D, and until indefinite, almost indefinite numbers of different antigens. If you take blood from those individuals, they will be containing antibodies from different B cells, therefore will be called polyclonal antibodies. If you've immunized this animal with against something, for example, you immunize the goat with the human IgG and C fragments, then still the antibodies, the serum will be polyclonal, but a larger fraction of those